have some interest in aging, but I'll start with a broad overview introduction to aging and what we know, what we don't know about it. And then I'll talk a little bit about some of our work on computational systems biology, machine learning approaches. So I'll give a few examples of some of the work we're doing and how I perceive the aging process and what drives aging. So, right, can you see my screen well?
Yep,
All good.
perfect.
All right. Great. So, as I said, I'll start with a brief introduction to aging. I'm sure you're familiar with the process of aging. That's one of the big advantages of working on aging is that everyone is familiar with that. Um, and so, but I mean, of course, there's more than meets the eye about aging, and I tend to have quite a broad definition of aging. So, I mean, these are some Czech centenarians, as I'm sure you'll understand. But the point is also that there's a lot of changes that occur with aging. There's cellular, molecular, physiological, functional, pathological changes. So, as I was saying, I tend to have quite a broad definition of aging as a progressive deterioration of physiological function accompanied by an increase in vulnerability and mortality with age, which I think encompasses the process of aging, which is deterioration, increased degeneration, increased vulnerability, increased mortality with age, and of course, increased incidence of various aging diseases, cancer, type 2 diabetes, cardiovascular diseases, neurodegenerative diseases, etc. So, um, and one of the big problems I think we have in the field is that we don't have a good understanding of why we age. We have a very poor understanding of the drivers of human aging, or even in model systems. We have a number of hypotheses about why we age, reactive oxygen species, epigenetics, mitochondria, senescent cells, loss of proteostasis, telomeres, DNA damage, mutations. There's a lot of ideas that have been around for, in some cases, decades, but we still have a poor understanding of why we age. It's perhaps not surprising because biology is complicated. We don't have a great understanding of many other facets of biology and many other complex diseases. But I'll come back to why we age in a little bit because I think when you look at the process of aging and what advances have been made, clearly the big advances in the recent decades has been at the level of manipulations of aging, um, particularly genetic, but also pharmacological, dietary manipulations. I'll mention more genetic manipulations because they tend to have a bigger impact, but I'll briefly touch upon pharmacological manipulations in a little bit. And I think this was really revolutionary for those who are not familiar with the history of the field, because if you look at the literature in the 60s or 70s or even 80s, people would say, it's not possible for one gene to regulate aging. That's not going to happen. It's going to have to be multifactorial. You're going to have multiple genes that to manipulate aging. And that's actually, at least in animal models, that's not true. We now know that you can manipulate aging with single gene manipulations. So the first works on this was, H1, which was identified by Tom Johnson and colleagues in Colorado, in C. elegans, in the roundworm C. elegans. And they were the first to show that, a single gene difference, they didn't know what the gene did, so that's why they call it H1. But that single gene variant has a significant impact on aging. So that was quite revolutionary. And of course, since then, there's been a lot of work. Cynthia Kenyon, Gary Ruvkun, who won a Nobel Prize last year, and many others have really shown that we can, to some degree, manipulate aging in animal models. And I always found this fascinating. The fact that you can tweak a gene and alter the aging process in animal models, I think it's quite incredible. And so back when I was a PhD student, I started this collection of databases and tools called the Human Aging Genomic Resources to help researchers study aging at the genomic level. And since then, these have grown to be the benchmark databases of the field with many visitors and users and citations. And I mean, if you're interested in the biology of aging, I would also um, uh, plug in our biology of aging news that I maintain on Facebook and Twitter. So whatever, I used to send it to my lab. Whenever I saw an interesting paper on aging, I would send it to the lab. Then I thought, well, maybe other people would be interested. So I started this pages on Facebook and Twitter that essentially just articles I find interesting. But anyway, I digress. Going back to the genetics of aging, so back when I was a PhD student, I started this database called GeneAge Database of Aging-Related Genes, which encapsulates really how much we've advanced in the field. And I think it's really quite impressive. I mean, what the latest version of our database, what we call the gerontome, it's quite massive. So we now know of over 2,000 genes in model systems that when you manipulate them in some way, you silence them, you knock them out, you overexpress them, it has a significant impact on aging or longevity. So, I mean, in C. elegans, we now know of other actually age one alleles, other genetic variants of age one that can extend lifespan by tenfold. And I think that's really incredible that you can tweak a single gene in an animal and make them live 10 times longer. And even in mice, of course, mice are more complex. Mammals like us, but even in mice, you can tweak a single gene and increase lifespan by up to 50%. So I think that's quite revolutionary. Of course, we don't know if this applies to humans, but at least in animal models, we can at the genetic level manipulate aging to a certain degree. Now, of course, the mice will still age, they will still die, they'll still develop age-related diseases, but they live longer and they are healthier for longer. You can also, I think I have a photo. Yeah, you can also accelerate aging with single gene manipulations, or at least some facets of aging, or most facets of aging. These are two mice of roughly the same age. The one on the right has a mutation, I think, is ERCC1, which is DNA damage response. And they have a short lifespan and develop phenotypes resembling accelerated aging, like gray hair, their eyesight is going worse. They have this, you can't see it very well, this curvature of the spine. So you can accelerate aging, not everything about aging, but at least a majority of the aging phenotype by single gene manipulations, and you can also retard it. In terms of retardation, the major pathway associated with aging, as you may be aware, is the growth hormone IGF-1 signaling pathway. So these are two mice of the same age. The one in the bottom has a mutation in growth hormone signaling, which, well, growth hormone, as the name implies, makes you grow. So if you have a mutation that disrupts growth hormone signaling, you have these cute little mice that they live a long time. They up to 50% longer than normal, and they're quite resistant to cancer as well. So they live longer, they are healthier for longer, and they will still age and die, but they do so at a later age. So extrapolating to humans, it's like making a 70-year-old having the health of a 50-year-old. So at least in mice, that's possible. What about humans? I mean, quite a few examples. I mean, obviously, we cannot do genetic manipulations in humans, but we can look at natural variants or natural mutations, naturally occurring mutations. So, this is the classical example of accelerated aging in human beings, a disease called Werner syndrome. As you can see, patients look older than a chronological age. They have an early onset of a number of diseases. They have a shorter lifespan. I think they live like late 50s. It's not a perfect phenocopy of aging. They tend to develop some age-related diseases earlier, but not all of them. So, for example, they don't tend to develop early onset of neurodegenerative diseases. So it's not perfect accelerated aging. It's a phenocopy about half of the aging phenotype. We also know from studies of centenarians that genetics plays a role. I mean, this is Jean Calment, who lived to be 122. I think she's a great example because she was very fond of chocolate and wine, and she smoked most of her life. So, she's not really the healthy lifestyle you'd expect from the oldest human being on record. So I think she's quite remarkable in highlighting really the impact of genetics. And we know that from other studies. I mean, when you study centenarians, I mean, there's no lifestyle factors in common. There are centenarians who have very healthy lifestyles, and there are centenarians like João who don't have very healthy lifestyles. However, it tends to run in a family. So there's been studies of heritability of longevity, and it's about 25%. So the genetic component of longevity, human longevity, is about 25%. Um, However, it increases with age. So what it means is that I mean, what I usually say is that if you want to be a healthy 70 or 80-year-old, it's mostly down to your lifestyle. But if you want to be a centenarian, like Jean Calment, or super centenarian over 110 years of age, is mostly down to genetics, mostly down to choosing your parents and grandparents well. So that's kind of what we know. Unfortunately, we don't know the specific genes involved yet. We don't. I think while there's been a lot of advances in the genetics of aging in model systems, um, lots of manipulations, lots of genes, hundreds of genes associated, over 2,000 in across model systems, our understanding of genetics of human longevity remains relatively poor.
Pedro, can I ask you a question about the
Yes.
so-called blue zones and whether there's a genetic difference in those demographics in those populations, or is it more lifestyle and environmental, in your opinion?
Not as far as I'm aware. I mean, there has been some studies in southern Italy, Sicily, and Sardinia. None as I'm aware. So it's mostly down to lifestyle. I mean, there's been some questions about the blue zones as well. Where it's more of a myth than than an actual effect. I think there are some areas like so. As far as I know, the longest lived population is in Okinawa in Japan, which is this island south of Japan. Well, it's more than one island, but this so that's the longest lived population, and they seem to have a very healthy lifestyle.
Hmm.
Um, you know, Almost like low calorie intake, low amounts of processed foods, and so on. So that seems to be mostly down to lifestyle. But definitely genetics of longevity runs in some families, and I think that's also important. But whether it's the same genes in long-lived
Hmm.
Sicilians and Okinawans, Portuguese and Americans or different areas of America, we don't know.
Hmm.
I think that's still a big issue.
Hmm.
Um, so the point is that there is a genetic component to aging, at least in model organisms, and there is some genetic component to human longevity and presumably aging. Although we don't know well the genes involved. And I've always found it quite fascinating because the genome is quite big, 3 billion base pairs, 20,000 protein coding genes, God knows how many non-coding genes, but very subtle changes in the genome can have a very big impact on aging. As shown in Werner syndrome, as shown in centenarians, as shown in model systems. So I think that's quite fascinating, and I'm a biologist, so I'm a cell biologist by training, actually. But well, my mother, she's retired now. She's a maths teacher. My dad works in computer sciences. So I think to really understand biology, to really understand complex biological problems like aging, really we have to take a systems approach. So I see biology as a information/mathematical problem, and given all of the technological advances in DNA sequencing, for instance, and DNA and omics data generation, there really has been this shift from data generation to data analysis. Um, so I think that's quite important. And so our lab reflects that. I mean, I lead the Genomics of Aging and Rejuvenation Lab at University of Birmingham in the UK. Um, Broadly interested in understanding why we age, what's the molecular, genetic, and cellular processes of aging. And we employ a combination of approaches, which I'll very briefly, I mean, I won't touch into a lot of methodological details, but I'll briefly touch on a couple of different aspects that we work on. So we focus on aging, particularly from a genomic perspective. I'll briefly touch upon pharmacological approaches, and I mean, interested in understanding why we age and how we can manipulate it. Do some evolutionary analysis and long-lived animals. Do some work on complex diseases like cancer. We do some cell work as well, and we do collaborations on animal works, like Nick and mole rats. We do with collaborators. And I'm quite interested then in computational approaches and conceptual models, which I'll touch upon today as well. Um, so 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 what I'll do is I'll give a brief overview of some of the research that we've recently or done or are ongoing in the lab without going into a lot of methodological details. I mean, most of it is preprints or has been published. So, but happy to discuss further in more detail. Um, I suppose from a conceptual perspective, my point, which well I mentioned already, is really understanding aging. Aging is it's a complex process. It arises from interactions of multiple genes with each other and with the environment, and so we need to take this systems approach to try to understand how all of these connections determine a phenotype, how the genes interacting with each other in the environment, how they modulate the process of aging, how they determine longevity. Um, so that's conceptually how I'm interested in tackling the biology of aging. And so let me show you a couple of examples. I mean, so let's start. This is something we did some years back already, but I mean, it's essentially we did this systematic analysis of the gerontome. So we did various different analyses of gene age, basically. But one of the major things was trying to classify what are the major pro and anti-longevity pathways. So you can classify genes as pro and anti-longevity based on whether they extend lifespan and whether you're upregulating them or you're knocking them out or silencing them. You can determine if they're pro or anti-longevity. And then from different model systems, we can identify in an unbiased way, what's the major pathways associated with pro and anti-longevity. And I mean, most of them are not surprising. Pro longevity, you have cell cycle regulation, including p53 signaling. Anti-longevity, you'd have growth hormone regulation and IGF-1, as I mentioned, the major pathway that modulates, that slows aging in mice when you knock out or you reduce growth hormone signaling. In flies, hypoxia activation, and anti-longevity, again, it's IGF. I mean, flies don't have growth hormone, but they have other neuroendocrine pathways like PI3 kinase, further downstream, and IGF. In worms, you have autophagy as pro-longevity, and then mTOR signaling and oxidative phosphorylation as anti-longevity. So the point is that, from an unbiased perspective, you can look at this. What are the major pro and anti-longevity pathways in model systems? I mean, we did a lot of other analysis in this, but I won't go into that. We do, I suppose, a lot of network analysis and in silico approaches. I think the point is trying to understand the whole from its parts, and we've done that in different contexts. So, I mean, we try to predict pathways that are associated with aging, as I show you, and trying to identify also new candidates, which we've done for aging. More recently, we did this for cellular senescence. So we published, so again, I'm not going to go into detail, but we built a network of cell senescence genes. So senescent cells tend to define, what's the definition of, I tend to define senescent cells as cells that should be proliferating but are irreversibly growth arrested. That's my definition, but there's a whole host of senescence phenotype, like this is actually from my PhD. I did my PhD in cellular senescence. The senescent cells tend to be morphologically quite different. You have some long cells, you have some big cells. So there are particular markers as well, like P21, P16, cell cycle arrest. So basically what we did is derived a list of a few hundred genes associated with cellular senescence based on genetic manipulations in vitro, and then we built a network of this, and then we identified various pathways. We predicted new hubs, new regulators of cell senescence, which we tested experimentally in vitro. And then we also looked into whether our signature of cell senescence occur in human tissues using human gene expression data, which we showed for some tissues, but not all. I would say not even most of them. So in some tissues, more than expected by chance, you do see signatures of cell senescence with age, but not most of them. So, interpret that as you wish. I mean, my own take actually. So, bit of a side note on cell senescence is that I think first of all, senescent cells are different types, so there's quite a lot of heterogeneity in senescent cells and different markers. That's important to keep in mind. But at least according to some markers, some senescent cells are important to us. They're not like cancer cells. I mean, cancer is very, well, if you have cancer cells in your body, you want to destroy them. I mean, I think that's very clear. That's unambiguous. Senescent cells is not like that. And there's quite a lot of studies now that show that if you destroy senescent cells, you can cause health issues in mice, even in human beings. There's a lot of, well, in mice, there's a lot of elegant genetic manipulations showing benefits of senescent cells, also detrimental effects. So I think one of the big I mean, I did this perspective last year, and one of the big questions to me is still which senescent cells and in which contexts, on which levels are beneficials and which are detrimental, because that's really important for us to determine when to target them pharmacologically, for example. So cell senescence to me is still a bit of a minefield. I mean, what exactly its functions, which we know it has some, and how is it detrimental during aging, and exactly when and how we should target them. And so, but I digress a little bit. Going back to computational machine learning approaches, so we do a lot of, as I said, we do a lot of candidate gene prediction, for example, using computational analysis, using machine learning / AI. We do a lot of supervised machine learning prediction, so essentially trying to predict new genes or new compounds associated with longevity / aging. I mean, we did this review, but let me, oops. Oh, I went back to the end. Okay, so sorry for that. But so let me go back to where I was. So we've done this quite recently in the context more of pharmacological approaches. So in addition to genetic manipulations of aging, we also know of a lot of compounds and drugs that can extend longevity. So we started. This is more recent. We started some years ago. We started this database called the Drug Age Database of Aging-Related Drugs, which now has over 1,000 drugs and compounds that extend longevity in model systems, which is quite remarkable. And longevity pharmacology has exploded. There's a lot of biotech companies working in this space, as I'm sure you're aware. I mean, one thing I would just caveat here is that the lifespan effects of drugs and compounds are relatively modest compared to genetic manipulations. So I think the most we can extend lifespan in mice with a drug or compound is about 15%. At least, well, there might be some obscure paper showing more, but something that's reproducible, that's about 10-15%, while the genetic level is up to 50%. Which makes sense because, of course, a biological, a gene mutation it has a much greater biological impact than a pharmacological one. But it's important to keep in mind that the effect sizes of drugs and compounds is smaller than genetic manipulations of aging and longevity in model systems. But on the other hand, it's still very exciting. I mean, if we could discover a drug that extends human lifespan 10-15%, that would be incredible. Even 5%, that would be amazing. If we could slow down aging, even if slightly, that would be remarkable and have massive healthcare, social, economic benefits. And so we've been doing some approaches on machine learning on this, so trying to predict new drugs associated with longevity with machine learning. So what we do, I mean, essentially a classification algorithm. So we have different compounds that extend longevity. We look at their, it's not showing here, but we look at their chemical characteristics. We look at their targets. We look at the pathways they target. We look at which aging-related genes they're associated or interact with. And then based on that, we can predict new longevity compounds, those that have similar characteristics to existing longevity compounds. So we've done this a couple of times. We have a preprint on this, and we published a paper 2023 now, of making these predictions of longevity. The more recent one, we actually look into mammalian data. So we started this in C. elegans, which we validated experimentally, which I'll mention in a second. But we're also doing using mammalian data, including in the goal is to use compounds that are already used in a clinic for drug repurposing. I think that's kind of one of my thinking behind it is there something already being used in the clinic that we could reuse that would have medical applications in broader age-related diseases. So one of the compounds we identified, we initially in C. elegans, but I'll show. So it's a compound called rilmenidine. Started a few years ago, so it's a long story short, but basically we identified as part of a in silico screen for longevity drugs. And what caught our attention is that rilmenidine is already used in the clinic. It's an oral antihypertensive drug. It has moderate side effects. We know the mechanism is targets these imazidulidine receptors. And so we did some experiments in C. elegans. So we just fed worms rilmenidine, and we see a dosage-dependent longevity effect, which is what you see here. So this is survival, and this is days of adulthood for C. elegans. So in black, you have controls, and then you have dosage-dependent effect. So 200 micromolar is about the sweet spot. More than that doesn't extend lifespan so much, or even doesn't expand lifespan at all. So we show it extends lifespan in the dosage-dependent effect. It extends lifespan at 200 micromolar, whether you feed animals early in life or late in life. You still see longevity effects. So what we're doing now, actually in collaboration with, Vadim Zgladyshev, also in Boston, we're been looking further into mice treated with rilmenidine. So we showed that they exhibit similar transcriptional changes in mice that other life-extending interventions. So mice fed rilmenidine, we do a gene expression study. They exhibit similar profiles, which is encouraging. And now we're trying to do a lifespan study as well. And ideally, I would like to do even a clinical trial, given in patients, to see if we have other benefits besides treating hypertension, of course. So that's something that, trying to get funding for that, trying to apply for some grants to get funding to do that. But this is, I think, is a good example of going from an in silico approach, computational analysis, to identifying a promising compound, testing it in model systems, testing an invertebrate model, doing some experiments in mice, and hopefully we can take it forward. So the other broad topic we're doing a lot of analysis is more on an evolutionary genomics approach. So as I'm sure you're aware, the cost of DNA sequencing had dropped dramatically in recent years, last 20 years or so, thanks to advances like this one, like different sequencing machines. And so we've been doing a lot in a different topic, which is more on comparative biology. So something else always been fascinating to me is the variety of lifespans across mammals. I mean, I love animals. I used to have lots of pets when I was a kid. So I used to have hamsters and painted turtles and, lots of different animals. But of course, a hamster doesn't live very long. Mice or rats live about 3 or 4 years maximum. On the other hand, monkeys can live over 40 years, elephants 70, humans, as I said, 122. And at the other end of the spectrum, you have whales that can live over 200 years. So there's, even if you take very good care of a mouse, it will still age like 25 times faster than a human being, which to me tells me that, the whatever differences between lifespan in mammals, they're encoded in the genome. It's not environmental. It's not the diet. It's genetically determined. So the lifespan, the aging pace is genetically determined. So that's something I've always found fascinating and been trying to understand this. So we've done quite a few analyses. I'll only, again, for sake of time, I'll only mention a couple of projects we've done, namely in studying long-lived cancer-resistant species. One of them you probably have heard of already is the amazing naked mole rats. I mean, we're interested in these animals not just because of their obvious good looks, but because of their cancer resistance. So they can get cancer. There's been a few cases now, but over 1,000 animals kept in captivity. There's only been a few cases. They seem to they live over 30 years, which is quite remarkable. So they're rodents. They're small animals, a little bigger than a mouse, smaller than a rat. And yet they live 10 times longer than mice and rats. It's quite remarkable. So we've been trying to understand, I mean, what are the secrets? We've done different analysis on them. And the other animal we've also been focusing in is the bowhead whale. I think this is even more incredible animals because they are the longest lived mammal as far as we know. So bowhead whales have been estimated to live over 200 years. I mean, this is it's a biochemical assay on the racemization ratios of the eye lenses. It's a paper in the 90s from a group in Alaska. So it's not incredibly accurate, but I mean, there's also been some observational studies, there's some anecdotal evidence, and when you put it all together, I mean, I think it's clear they live longer than human beings, and they live longer than human beings in the wild, I mean, without doctors. And if you think about cancer as one rogue cell that just proliferates uncontrollably, then these animals, they might have tumor suppressors that we lack, right? I mean, otherwise they would die early of cancer. And so we've also sequenced the genome of the bowhead whale, and we've done various analyses of trying to identify genes associated with longevity and disease resistance of these animals. So I mentioned it for the naked mole rat and for the bowhead whale, we created these genome resources that are freely available. So we make all the data freely available online, and we're trying to do we identified some candidate genes under selection in these animals that could be responsible for their longevity. I mean, my dim experiment would be to take some genes from the bowhead whale and put them in mice, or at least or replace the mouse homologs for that, and see if they live longer or cancer resistance. That would be my dream experiments if I had the funding for it. But the point is also that we make all these genome resources available, and I see these long-lived disease-resistant species as complementary. So I see mice and as a model of disease like cancer, and I see naked mole rats and bowhead whales as models of disease resistance and longevity. So I see them as complementary. I see them as models to identify processes and mechanisms and genes that protect against disease and protects against aging. And at least evolutionary, we can identify some candidates for that. So my goal is that the broader biomedical community also uses these non-standard model systems. So in the last 5 or 10 minutes, I'll go back a little bit to why we age and mention a little bit about some of my ideas on this, which I think there's some overlaps with the work that you guys are doing as well. So as I said before, we have a poor understanding of the drivers of aging, although there's a number of hypotheses. DNA damage, telomere shortening, oxidative damage, protein damage, and so on. So those are the hypotheses. But there is very limited empirical evidence. Now, maybe reasons for that. Maybe we don't have the tools to test this hypothesis appropriately. I mean, that may be a limitation. We just don't have the technologies to do it. That's one possibility. The other possibility is there may be other facets to aging that we are not really looking into. So one of the things that have always fascinated me from a comparative biology perspective is that there's a very strong correlation between developmental times and growth rates and longevity. So this is from an analysis we did some years ago, but basically each of these dots is a different mammalian species. So, the bowhead whale would be here, the longest lived animal. So what you see here is longevity on the y-axis, and on x-axis is time to maturity. In this case, time to sexual maturity, which is an approximate. And what you see is a very strong correlation, which you see across different classes and so on. So the point is that the time it takes to reach maturity is a very strong predictor of the time, how long animals are going to live, of a given species are going to live afterwards. So I've always found that fascinating. And as I'm sure you're aware, I mean, one of the I think fascinating discoveries in the biology of aging in the last decade has been this methylation clocks from Steve Horvath and others. This observation that you have epigenetic clocks, you have these epigenetic changes that tick, from shortly after conception all the way to old age, which I think is fascinating. I mean, if you ask people 20 years ago or 15 years ago if this was possible, most people would say no. I would have said no. No, there's no way there's like some methylation changes that you see all the way from shortly after conception all the way to old age. No, there's going to be differences between cell types and differentiation and development. No, you're not going to see that. But we do. I mean, we do see them, and now they're highly reproducible. So I found that fascinating. So, and another thing that's fascinating is that it also works across species. So, it's not just, I mean, it was initially identified in humans, it's been shown in mice, but it also works across species. So, I mean, it's not the same CPG sites, but you also have CPG sites that change with age at different paces, of course, in different species because they age at different paces. But it's the same changes. Again, I think it's incredible. So one idea I've had for quite some time is this idea that, okay, maybe we don't age because of damage, but because of more about information changes, more about genetic programs. So maybe we have some developmental programs early in life that continue late in life as a form of antagonistic pleiotropy. So, you'll have some changes early in life. Um, you know, um, for example, changes in thymocytes in the thymus. Your thymocytes, they turn into fat when you're about 3 years of age. So maybe you have these changes and it's genetically regulated, it's not damaged, and it's optimized for reproduction because you want, the amount or the percentage of thymocytes or differentiated sites to be optimal for reproduction, but then it just continues later in life and becomes detrimental. Or you have something that increases. You have some tissue that keeps growing and becomes detrimental later in life, um, which is a different way of perceiving the process of aging from traditional damaged or error-based theories. And so 2 years ago, I put forward this hypothesis of aging as a software design flaw, which kind of brings together these two concepts. One of aging as antagonistic pleiotropy or as a programmatic process, um, and but one that runs in the epigenome. So the core idea is, okay, so maybe aging is not molecular damage, or I think there is some aspects of molecular damage associated with aging, like mutations in cancer, but it's rather a product of gene programs or programmatic processes that run in the epigenome. So that's essentially that's the idea. So what this means now, if you think about this, is the non-controversial part of the hypothesis, which is many others have mentioned this, is that, okay, maybe we have the genome, we have that encodes, you could see it, a very complex software program that then runs in the epigenome and determines different cell types, for example, and different life transitions across time and space. So that's not controversial. I think most biologists would agree to have. We have this genetic software program, and then different aspects of the epigenome run differently across space and time, and biological space and time. The idea is very simple, is that maybe this continues later in life and becomes detrimental. So again, we have all these programs, subroutines that run that are beneficial early in life, but maybe some of them, not all, but some of them continue later in life and then become detrimental. That's essentially the hypothesis. And this runs in the epigenome. So the differences you see and is mostly at the epigenome. Then, of course, there will also be gene expression changes and information changes and cellular changes. But the point is that this running of this developmental program would be reflected in the epigenome in different cellular epigenetic states. So that's essentially the hypothesis. It fits some of the observations. It fits, as I said, the observation that that there is a very strong correlation between age of sexual maturity and lifespan in mammals. This is just like an updated version from some analysis I did previously, and it fits some of the major pathways that retard aging in model systems. So like dietary restriction, like growth hormone IGF inhibition, and like rapamycin. So all of this impact or slow down growth and development, and I would argue then is via this retardation of the developmental software that then retard aging as well. So that's essentially the hypothesis. Now, I think in the context of, for example, cell reprogramming, for which there's a lot of interest, you could see it as a reprogramming, a resetting of the epigenome, where you have this epigenetic states that are by inducible potency or nuclear transfer that are reset and go back to factory settings. Let's call them that. That's kind of the way I see it. And so, to summarize, I do think molecular damage plays a role in some aging phenotypes, like mutations. We've done some work on cancer, but I didn't have time to go in through today. But I think there's overwhelming evidence that mutations are one of the drivers of cancer. Might not be the only one, but they occur in cancer and are one of the drivers of cancer. So, and cancer is an age-related disease, so molecular damage plays a role in aging phenotypes. But perhaps in that's not even the prevalent process. Perhaps we need to start seeing aging as an information problem and not just a damage, not just a stochastic damage accumulation process. And hence, we need to decode our genetic information and epigenetic information to understand the process of aging. So in a nutshell, that's this idea of seeing aging as a software design flaw. And I mean, I'm essentially done. So if you have any questions, but let me just summarize. So aging remains a mystery of biology. We don't understand what are the drivers of aging, what molecular, cellular, mechanistic drivers of aging. On the other hand, network analysis can lead to new insights, new pathways associated with aging, new genes, like we did for cellular senescence, new drugs. So we really need this computational analysis. I mean, I know you do some of this work already, but we do this computational systems, machine learning approaches to really tackle the complexity of biology, which we still don't understand very well. I mentioned also our work on long-lived species to find genes that protect against aging, which is complementary to traditional approaches of using disease models. I mentioned our work on rilmenidine as a new geroprotector emerging from our network and machine learning computational analysis. And lastly, I told you about software design flaw hypothesis. And I mean, with that, so this is where we're based.
It's not just me, is it?
No, I think you froze.
Okay.